A gene is a segment of DNA with a specific sequence of nucleotides that contains the information for making a protein. The gene is bound on either end by two sites, the promoter site and the termination site. Transcription begins when an enzyme called RNA polymerase binds to the promoter site. As RNA polymerase moves along the DNA, the two strands are separated. It is important to note that whenever a protein is being made, only the segment of DNA that encodes for that protein is separated. The entire length of DNA is not separated. Complementary RNA bases bind to the unpaired DNA bases on one of the exposed strands and are then attached to one another by RNA polymerase. These individual RNA bases are found floating freely in the cell. Recall uracil is the complementary nucleotide to adenine in RNA. When the RNA polymerase reaches the termination site, the separated DNA strands bind to reform the double helix, and the new RNA molecule moves away. This RNA is a copy, a transcript, of the information contained in the gene. The machinery which reads the mRNA and synthesizes the protein is called the ribosome. Ribosomes have two subunits, one large and one small, which assemble on the mRNA. The large subunit has two binding sites for another type of RNA called transfer RNA, or tRNA. tRNA is the molecule responsible for relating a codon to its specific amino acid. tRNA has an amino acid attached to one end, which will be used in the assembly of the protein. The other tRNA end has a 3-nucleotide sequence called the anticodon, which is complementary to the codon on mRNA. The ribosome assembles on the start codon of the mRNA AUG. tRNA with the anticodon UAC and the amino acid methionine binds to the AUG codon. This tRNA is in the P site of the ribosome. The A site is open for a second tRNA whose anticodon is complementary to the next mRNA codon. Once the second tRNA is in place, the first tRNA's amino acid, methionine, binds to the second tRNA's amino acid. The first tRNA then leaves the P site, and the ribosome slides down the mRNA to reposition the second tRNA in the P site. You can see that this tRNA contains the growing polypeptide chain. A third complementary tRNA enters the now empty A site. The polypeptide chain on the P site tRNA is transferred onto the amino acid in the A site. Again, the P site tRNA leaves and the ribosome slides down the mRNA. This cycle continues as the ribosome moves down the mRNA. The amino acids are stitched together to create a polypeptide chain according to the order of the codons on the mRNA. When the ribosome reaches a stop codon, a release factor binds to the A site. It carries no amino acid. This release factor signals the ribosome to release the newly synthesized protein. The ribosome then disassembles from the mRNA and moves on to find another mRNA molecule.